Dear friends and colleagues, a very good morning to you all. It gives me immense pleasure today to be introducing Dr. Sundeep Dugar to this webinar about mitochondria and consequences of its depletion and dysfunction. To be able to introduce Sundeep to a knowledge session is in itself a big privilege for me. Sundeep is a drug discovery scientist with over 4 decades of R&D experience. In this field, to be a part of a team that has discovered a drug is a rarity. But to be an inventor on 2 life-saving drugs is a feat that very few can boast of. With his enthusiasm for science and knowledge, Sundeep could have easily been a professor at any of the leading universities. But he chose a career that is committed to impacting lives. Sundeep has started numerous companies and advanced multiple drug candidates towards clinic. Sundeep was the founder of Sphera, responsible for creating the very company that we speak from, and also my mentor for as long as I can remember. Along with me, there are a few other people in the room who can very proudly say that Sundeep has been a teacher, and we are super thrilled to host this session. Sundeep is currently serving as the founder CEO of IM Therapeutics, founder and CSO at Biogenesis Therapeutics, founder and CSO at Neurogenics, and founder and CEO at Verify Therapeutics. As Sphera partners with Sundeep's newest venture, Blue Oak Nutraceuticals, we aim to bring some of the leading experts in mitochondrial health from the world stage to India. Today's webinar will involve a presentation, a short break followed by an interactive chat, and an audience Q&A session. If you have any questions, please feel free to submit it in the chat box, and we will take as many questions as possible. In line with this, and without further ado, I invite Sundeep to commence his talk and share his years of knowledge and research about mitochondria. Thank you, Sundeep.
Thanks, Abhinav. I think that introduction was good to hear, but not necessary, but appreciated. And thank you all for joining us this morning. So what I'd like to do is start by taking you back right at the beginning of life itself. Because the story of the mitochondria starts not just hundreds of years ago, but billions of years ago. So let's take a journey through time and get back. Question is, what is the mitochondria? What does it do? And what is its role in human life itself? So the theory of endosymbiosis was proposed by Lynn Margulis in the 60s, which states that some of the organelles in today's eukaryotic cells were once prokaryotic microbes. And as all the cells in our bodies are eukaryotic cells. So they contain the microbe, subsequently named the mitochondria. And in those days, about 2 billion years plus ago, eukaryotic cells like amoeba got nutrients by phagocytosis, by engulfing other single-cell species on planet Earth. And some of these phagocytosed prokaryotic cells survived and developed a symbiotic relationship with the amoeba-like cell. Hence, instead of getting destroyed and eaten up, they survived. And in a simple form, the product of the prokaryote became fuel for the eukaryote cell. And that led to the formation of the modern cell as we know it. The second endosymbiotic effect brought in the cyanobacterium into the system that created chloroplasts, and the original bacterium became what we call today is the mitochondria. And then the host cell provided these endosymbionts a safe place to thrive, and the endosymbionts worked and provided energy for the host cell. An interesting accident also happened around this time, which often is not talked about, and what we call is the great oxygenation event. photosynthesis started to come about on planet Earth. Oxygen levels started to rise. There's an interesting perspective on this. So think about this. 2 billion years plus ago, before that, Earth had no oxygen. It was a reductive atmosphere where life could not exist. Cyanobacteria did, but not the life we know of today. As photosynthesis evolved and oxygen levels started to go up, we had this potential of an endosymbiosis happening. It is possible that if this does not happen, the oxygen levels on Earth will continue to go on and would reach toxic proportions to prevent life from evolving. So there could have been a balancing act that oxygen comes up, endosymbiosis is happening, and the presence of that oxygen allowed this endosymbiosis to take hold because now you have oxygen, and at the same time, it started to consume the oxygen to level it off. It's possible that a lot of the exoplanets out there that we identify today that have organic material actually never evolved life because there were enough toxicants that were created that honey. Here's a pictorial thing. So on the left, I think if I have the pointer, is there a pointer somewhere? Thank you. All right. Oh, it won't work on that screen, unfortunately. I'll use the pointer here. Hopefully you can watch that. So here is your ancestral eukaryotic cell. Here you have the bacterium. You have the first evolutionary event that creates this endosymbiosis process that leads to the animal cell lineage. Then a photosynthetic bacterium comes in that leads to the plant. What that means is that every living species on planet Earth have mitochondria. You, me, animals, plants, everybody, okay? In essence, the mitochondria is a bacteria that lives in your body. And it has similarities to other modern bacteria outside of our cells, because like the modern bacteria, the mitochondria has its own cell membrane. So you have your cell membrane, and then the mitochondria have their own cell membrane within your cell, and they have their own DNA. So while we have learned through our lives that your DNA is in your nucleus, you actually have another set of DNA that is in your mitochondria. And I'll come to that in a moment. And mitochondria also reproduce like bacteria by pinching and splitting in two. Eventually, these bacteria, the mitochondria, lost their capability to grow outside their host cell. So they don't have, so the original bacteria lost 99% approximately of its DNA and maintains only about 1% of its original DNA. So it cannot survive outside the cell. But at the same time, if you remove the mitochondria from your cell, cannot make mitochondria. So there's a complete symbiotic relationship where they can't survive without each other outside in presence of this. And as I mentioned, all eukaryotic cells have them. So what is this mitochondria and where does it come from? It only comes from your mother. It does not have any paternal lineage. It only has maternal lineage because it comes from the egg. So in your cells, every cell, you have a nuclear DNA that has DNA from your father and your mother. And as generations go, that DNA gets diluted. So your DNA goes to 50% to your first generation, 25% to the next, and so forth. But the mitochondrial DNA stays the same. So it stops with the males and it continues with the females. So the women have won the first battle of the sexes, right? They control your life. So when you go home today, do thank your mother. okay? But this also has an implication on the evolutionary theory of life. We've been taught that genes are the blueprint of life. Let me give you a different perspective. I would even say that without the mitochondria, the genes cannot do anything. And it gives you an evolutionary explanation beyond the gene, and that is of metabolism and energy as a central organizing feature of life. Because without energy, the universe cannot exist. Imagine the sun not being there. The sun's purpose is to give us light and heat, two forms of energy. Without it, we cannot survive. And as things would have, energy and mass is constant in the universe. You cannot destroy it. Those are the two fundamental parameters. One can even argue that the interconversion happens, theory of relativity, as Einstein put forth, is what happens. That solid things are nothing but severely condensed forms of energy. Yeah? It focuses on biochemical networks rather than genes. What are the cellular, metabolic, biochemical signalings that are happening that leads to sustenance of life and health in life? And I'll touch on that a little bit. It focuses on phenotypic interactions rather than informational inheritance. Because remember, the lineage of your mitochondrial DNA is the same. But the idea is what that does is controls the phenotype that you present yourself to the world. And it's a view of evolution, hence, are valuable for expanding evolutionary explanations. It does not negate the role of gene and the genetic part of evolution, but I think it presents a compelling alternate argument of evolution on planet Earth. And as I mentioned, energy is the primal force and basic component of the universe. So is DNA a blueprint of the right? Let me pause it. Probably not. It is an essential component in life, but we have so long thought about the genetic basis and have ignored the mitochondrial component of it. And I'm here to tell you that the mitochondrial component is as, if not more important, than the genetic basis of life. Okay, mitochondria and health. Okay, it touches every organ of your body, right? Every organ, every cell except for one, the red blood cells. So mature erythrocytes do not have mitochondria, but every other cell in your body does. As a matter of fact, all of you are sitting here. If you move your hand, you blink your eyes, you scratch your face, even listening to me is requiring your mitochondria to provide you the energy to do so. Even passive actions like listening requires energy, because when the sound waves hit your eardrum, they promulgate the whole process that requires energy. So some facts about the mitochondria. It's an only component in your cell that can make more of itself when needed. It can fix itself when it's broken, and it gets rid of itself when it's not needed. Now that's very significant. And I'll remember those parts because as I go through the talk, you will understand why those three components are relevant to life and health. Okay? And it's the only thing that can do that by itself. It controls its own destiny. Okay? The cellular destiny, as I said, is controlled by a symbiotic relationship. But the mitochondria also dictate the cellular path or its function. Okay, such an important organelle. Yet there's one way to make more of it. So if you want to make more of it, the biogenesis, exercise is the only known physiological stimulus to make more mitochondria. So here you have an interesting issue, right? So important, but in the absence of physical activity or other activity, even cerebral activity, which is mental exercise, you cannot make more mitochondria. Okay, so all of you, after you're done today, should go out for a 10-mile walk, because that's healthy for you. You'll make more mitochondria. Okay? And if anything you get out of this, you get an understanding why you should exercise. Yeah, it's good for you. Here's the fundamental reason that you make more mitochondria. But it's also susceptible to destruction when you don't eat right. Pollutants in the air, and your lifestyle, stresses in your life, they all affect the mitochondria, and you will destroy mitochondria. And there's a singular way. It's called the formation of these permeability transition pores, where the mitochondria swell, they make holes, release cytochrome C, and the cell is dead. Now, what you eat, they take in the nutrients and turn it into energy. Energy is ATP. I'm not going to talk much about ATP, but singularly making ATP, to all the chemists in the room and outside in the audience, will appreciate that what you're doing, what this molecule is doing, or the organelle is doing, is taking two negatively charged molecules and putting it together. All right? That is against all the laws we have ever heard. Yet it does it effectively and very efficiently. So food you eat, you have lunch, dinner, those are nutrients for the mitochondria. It's the mitochondria that process this food that you eat and make ATP. All of us have it, of course. You have about 100,000 trillion of these in your body. That means you actually have more mitochondria than human cells. All right? About 25% of cell volume can be mitochondria. Now, here the numbers go crazy, right? An average cell uses about 10 billion ATP per day. All right? And imagine how many cells you have in your body. Okay? So each cell is making this ATP, putting two of these negative things together billions of times every day for all of your life, in every cell of your body. And what is the funny part is, the body cannot store ATP. It has to make it on demand at the time you need it. Okay? You nod your head, your neck muscles need ATP for you to nod your head. It'll make it at that instant. Now, if it doesn't impress you, I don't know what will. Just imagine this little organelle in every cell is making billions of ATP on demand in an instant, in a reaction that is almost impossible to do in a lab. Okay? If I were to take the amount of ATP you make per day in all of your cells, pulled it out of your body, it would be close to 150 to 160 pounds of it every single day. Okay? Now, the rest of the data is here that tells you about how much you can make per minute, and total is 10 to the power of 25, etc. But the proficiency of this process is so impressive, and it goes on without you even having to think and worry about it. So the least you can do is take care of it. Two things affect the mitochondria: the food you eat and the exercise you do. Very simple. It gives you a simple idea. So the next time you go out and put a food morsel in your mouth, think about it. Right? The next time you feel lazy and don't want to exercise, think about it. And that's all it asks. It says, eat, exercise, and I'll take care of you. Remember that. I'll come back to it in a little bit in terms of biogenesis and exercise, right? So I'll come back to that. So what this means in terms of health is that the mitochondria has associated through two genomes. okay? It has its own genome. And if it is affected or mutated, it presents with a disease. But if there's a nuclear genome that is affected, it affects the mitochondria as a secondary mitochondrial myopathy. Okay, so everything that happens in your nucleus, in your nuclear genome, the mitochondria sense it, they respond to it, and if that insult continues, you start to see the disease presented in humans. Okay, so that's the expression. So here you again, the disease pathologies that we speak of, most all that we know, and as a scientist, I'm not going to say all, but it's pretty close to that, will touch the mitochondria. Okay, so what are the diseases? Metabolic syndrome, diabetes secondary to prescription drugs. What is fascinating is over 70% of the prescription drugs are toxic to the mitochondria. Yeah? It's, to me, who's been in this industry, it's absolutely interesting that we do all kinds of toxicities on our drug development, but we do not do mitochondrial toxicity. When you take statins, it's toxic to your mitochondria. You take SSRIs, it's toxic to the mitochondria. Yes, we have low cholesterol, but we're destroying one of the fundamental components of your cell that keeps you healthy. Okay? And I put DOT because I can go from neurodegeneration, Parkinson's, kidney failure, heart failure, sarcopenia, and I could go on, and I could take the whole hour talking to you about all the diseases. Okay, aging, think about it, is the disease. You have the maximum number of mitochondria in your body at about age 20, 21. That's about when you stop growing, okay? Growth from day of birth to age 21, your body is undergoing internal exercise. Your bones are growing, your muscles are growing, your whole system is growing. And the body needs the energy. And it recognizes that as an energy and continues to produce the mitochondria you need to make it. At about 21, you stop. So your internal exercise stops. From then on, it's all downhill. You lose 10 to 15% of your mitochondrial content every 10 years, right? So by the time you hit 30 or 40, suddenly your muscles weaken up, your metabolism slows down, and you wonder if I eat one piece of bread, I gain weight. When you could eat 10 when you were 16, suddenly you eat one and you gain weight. Your metabolism is slowing down. So you start developing wrinkles, your hair turns gray, you lose your hair, your mental capacity goes down, mitochondrial energy is going down. And with that comes oxidative stress, and you also induce mitochondrial mutations. Because replication is happening so fast, when you have the bioenergetics, your cell is able to clear out the mutated mitochondria. As the cellular energy goes down, the cells have to make a choice where to expend that energy. And suddenly these mutated mitochondria start to accumulate, okay? And that leads to decreased autophagy, increased apoptosis, and decreased mitochondrial biogenesis, et cetera, okay? So think about aging is a long-term disease. And as I said to you, what happens when you make exercise? When you do exercise, you make more mitochondria. What does it mean? So that means when you exercise, in the center is the muscle, you make more mitochondria, and within the mitochondria are what we call cristae. So interesting about the mitochondria is that it has two cell walls. In the inner membrane of the mitochondria exist these cristae, undulating part, which is where ATP is made. And more cristae you have, the more functional the mitochondria is. Okay? So when you exercise, it does it. I mean, there's a lot of other things going on. I'm simplifying it only to the point of mitochondria, but of course, there are many changes remodeling, so many remodeling happens, including the Yamanaka factors, and all kinds of things are happening. But they're all interrelated. Okay? So let's focus on the mitochondria. It introduces mitophagy. What does mean is now with exercise, you have energy capacity to remove these mutated or dysfunctional mitochondria, right? So that exercise induces. And it enhances mitochondrial respiration. That is oxygen consumption, you burn more food, and of course, you lose weight, okay? So the question that I asked was, how? So this became an intriguing question, that exercise induces mitochondria. What are the biochemical pathways? Remember I mentioned to you about the biochemical processes that are important that induces more mitochondria to happen. I've told you that the mitochondria make more of itself. So you exercise and they make more of themselves. So here's a concept. If you have a cell that's dividing, it will make more mitochondria so that the two daughter cells have the same population. So that's mitochondria based on internal processes of cell division. But with exercise, you make more mitochondria without the cells dividing. So the machinery of mitochondrial biogenesis is different. The question is it? So as I went around and talked to all the people who could take, nobody could tell me why and how does the mitochondria make, or cells make more mitochondria when you exercise, or the mitochondria make more of themselves, whichever the way you want to look at it. So let's go a little bit deeper in it. One of the functions that happens when you exercise, that's the central arrow, is you create ROS, reactive oxygen species. Fair? And on the left side is that if you're exercising, you have better adaptive response. On the right side, you don't exercise, you have no adaptive response. But you make ROS. That's known. But here's the conundrum. Everybody tells you ROS is bad, right? Take antioxidants. That's the prescription for health. Yet the body's making ROS. That doesn't sit well. Why would the body, if exercise is good for you, why would the body make ROS, which we think is bad for you? It does not sit well. Which is where the equation of balance comes in. Because the difference between a poison and a potion is dose. How much? So the general theory is ROS is pathogenic. On the top right, mitochondrial respiration goes up, creates ROS, and everybody tells you all these other bad things happen to you. Lipid peroxidation, DNA damage, protein oxidation, la-di-da-di-da. So go out and get your vitamin E. So the balance of ROS is the key. There's the good and bad of ROS. In the middle lies the beneficial levels. If you have too much, it's bad for you. If you have too little, it's bad for you. So it's not that it's pathogenic in it of itself, it's the level that matters. Okay? ROS can also be made by bad food. If you eat a lot of sugar, you'll make more ROS. Okay? But that ROS is not through the signal of exercise. It's a separate signal. But this ROS is coming through a systemic requirement for the cells to increase their bioenergetic level, and that ROS is the part. Okay, but then what happens? I'm a chemist by training, right? So we believe in the molecular basis of science, right? So if there is a level of ROS that is good for you, what happens? Now, this takes you to a separate part of the story. So let's go over this briefly. Mitochondria are responsible for all initiation of all steroid synthesis in your body. They start the first step. Okay? And I'll highlight the two. The first step is on the middle, on the top, where they take cholesterol and convert it to pregnenolone. mitochondria do it. Without pregnenolone, your body cannot make steroids. Okay, so the mitochondria take cholesterol, convert it to pregnenolone, and then in the adrenal, the rest of the steroid synthesis happens. But then there's one last step that still happens in the mitochondria, which is on the right, where there's a hydroxylation step. And let's go with it. I'm not going to explain what that is to everybody, but there's two steps that the mitochondria control. One is at the start, and one is towards the end. Okay? Now, interestingly, both of these enzymes are cytochrome enzymes. Cytochrome enzymes are iron-containing enzymes. They have iron in them. And to all of you, iron has two oxidation states that generally exist, ferrous and ferric, Fe+2 and Fe+3, right? And that oxidation step is managed by ROS, because cytochromes need the oxygen for an oxidative process. So ROS can activate the two enzymes. Okay, so if we follow this theory, as you exercise, you make ROS. And when you make ROS, these two enzymes can get activated in the mitochondria. Okay, so now you have them activated. If only these two steps happen, remember, your muscle does not have the capacity to do steroidogenesis. So the mitochondria in your muscle that is exercising can only do these two steps, okay? And if it does these two steps, it will make this molecule called 11-beta-hydroxypregnenolone. Okay, step one, and the last step, that's what you get. So the question one asks is, what is known about this molecule? Because the theory would say, if I'm exercising, I'm creating growth, why am I not making this? So I went into PubMed, nothing known. No reported pharmacology of this molecule. But it would make sense that exercise should produce it. Anyhow, so this is where, and by the way, thanks to all of my colleagues at Sphera for the many years, we went through this journey together, and we made this here in Sphera Labs and tested it. Before I gave you the data, so this is the putative biosynthetic pathway. You go from cholesterol, it goes into the mitochondria. The first cytochrome, CYP17A1, is the one that makes pregnenolone. The second one is that puts that hydroxy group to make 11-beta-hydroxypregnenolone. And this is what happens. When you take exercise, you make ROS, goes to peroxide, that activates the two enzymes. But then ROS does something else also. It changes the ratio of NAD plus and NADH, which means it increases the oxidation level of the cell, where it can take hydroxypregnenolone and convert it to hydroxyprogesterone. Okay, so if this theory is correct, both of these should be relevant. Okay, and I'll get to that. So let me also say down the part that the fundamental hypothesis actually is supported by steroids acting as stress responders. Okay? If you have an acute stress, what is that? A tiger comes here. All right? Acute stress. Right? Or a king cobra is sliding on the thing behind you, right? Our stress levels shoot up and our cortisol levels go up. And you hit in the fight or flight mode. Okay? So either you're going to stand up and get ready to fight to survive, or you're going to run like crazy. Correct? So your body produces this cortisol to create the environment for your body to react to it. This is acute stress. Okay? Because it happened there and then it's gone. Think about exercise like a chronic stress. Because when you run, you're asking the body to produce more energy. And that's a chronic stress. You don't do it all the time. You don't do it episodically, but you do it regularly. Okay? So it's not extraordinary to think that is there a process that manages acute or non-acute stress? And that builds resilience for future challenges, such as muscle mass goes up, cognition may get better, mitochondrial dynamics. So the hormonal response systems to survive and adapt to stressors are there. Hence, a role as a mediator response to non-acute stress, such as exercise, growth would not be unprecedented. Okay? So there's precedence in our body for responding to acute stress. So there should be, it's reasonable to assume there's something for the non-acute stress. Is 11-beta-hydroxypregnenolone now the hormone of exercise? Now, good question. Now we have to see what the answer is. Okay. So we made this and we tested. And sure enough, we see a very significant increase in its effect in creating mitochondrial biogenesis. This is cells, and you can see that the activity is down at the femtomolar range. This thing is incredibly potent to induce mitochondrial biogenesis, which also makes sense because living itself is an exercise. Just walking is an exercise. And you don't need to have to produce lots of it to get the effect. So the body can produce very small amounts of it immediately and initiate in the cell that is exercising to deliver mitochondria. Now there's another part to exercise. You run and you lift weight, but your whole body feels good. Your mind feels good. Why is that? And I'll come to that a little bit later. Okay, so let's focus on the local effect, and I'll talk about the systemic effect just in a little bit. So, your body exercises, it produces hydroxypregnenolone. Here's the first data that shows that, yes, I can induce mitochondrial biogenesis in cells through this. This is the first time anybody had ever studied this steroid to understand its mitochondrial function. Actually, we're the first ones to understand its pharmacology or biology to begin with, because nobody has ever reported the effect of 11-beta-hydroxypregnenolone. So it makes more mitochondria, but the cell is such an incredible system. So think about this. You're exercising, your body's making ROS, correct? Now, the cell does not know whether this ROS is pathological or it is pharmacological. It doesn't know that, correct? So what the body does is it creates defense mechanisms alongside the mitochondrial biogenesis. And one of the aspects of this steroid is that the mitochondria make it, it comes out of the mitochondria, it goes into the nucleus. I'm not going to talk about that process today, but we understand how that happens. But this steroid also within the mitochondria protect the mitochondria in case the ROS gets too much. And the way it does it, and this is something we've published, is I told you, remember the permeability pores I mentioned to you early on? ROS can induce permeability pore. What this steroid does is within the mitochondria sits on one of the complexes, perhaps for another day, we'll talk about complexes, and protects the mitochondria by not allowing these pores to form. So what is it doing? In summary, it's made by the mitochondria, it goes into the nucleus, tells the nucleus, initiate the process of biogenesis in the absence of cell division, and stays back also to protect the mitochondria in case the ROS damage is uncontrolled. So you'd say, why should I make more mitochondria and start killing the ones I already have? So the nature is creating a balance that we make more, but also protect what we have, okay? And this steroid does this, and this is published paper, so we've published that. Let's go to the next part. So we show mitochondrial biogenesis, okay? What is the pharmacology of it? These are just a little bit of data. We could be here for all day if I went through the whole thing. This is just in HepG2 cells. And if you focus on the middle and the right one, what it does is it increases triglyceride consumption within the cell, and then it increases the level of hydroxybutyrate. That is telling me that the cells are burning fat, okay? Which is what the mitochondria likes. Fat is the best mitochondrial fuel because the ATP levels made by fat oxidation is much more than glucose glycolysis. All right? So these cells with more mitochondria have induced fat burning phenotype. This is some in vivo studies. On the left is a study in a cardiac ischemic reperfusion model, because when you have a heart attack, you have deprived your heart of the oxygen. So the mitochondria in the heart cell are now facing toxins and excessive ROS. So when the blood comes in back again, actually that's when the real damage happens. That's when the mitochondria die, because the levels of ROS skyrocket. And what we've shown with hydroxypregnenolone, that it manages that because it permits, remember I told you about mitochondrial protection. It protects the cell mitochondria and does not allow the cells to die, which is very critical because post myocardial infarction or a heart attack, you can now actually have less heart damage than you would otherwise. Hence, your outcome could be better. On the right side is lactic acidosis. Coming back the circle to exercise. Remember exercise I mentioned? So one of the reasons when you exercise and you get tired, or you can't exercise, or your muscles hurt, is because you build up lactic acid. And what is happening is that as the whole glycolytic pathway is going through, you make a molecule called pyruvate. Pyruvate goes into the mitochondria to create the energy for ATP synthesis. But if you pump more pyruvate and your mitochondria are not able to consume it, that pyruvate comes out as lactic acid, okay? So you have a certain amount of mitochondria and you exercise. You can run half a mile, but you can't run 2 miles because your muscles are not ready for it, because they don't have enough mitochondria to continue to consume the amount of energy demand you're doing. What hydroxypregnenolone does, because it increases the mitochondria, it allows you to consume that pyruvate so you don't make the lactic acid, and that is a direct indication of increased mitochondrial function. So these were rats that were made to swim for 45 minutes. They were not very happy, obviously. They didn't tell me that, but nonetheless, their lactic acidosis was significantly reduced. So then, the key question is, do you make it? Okay? So this again was great studies done here at Sphera. Great group of people that have supported a lot of this work over the last 14 years that we've been at it, 16 now, I guess I lose count after a while. And this study was done in rats, and what you can see is the key, that on the bar graph you can see post-exercise within 5 minutes, the levels of 11-beta-hydroxy progesterone. And I'll come back to that, okay? Remember I told you there's the pregnenolone and the progesterone is outside of the mitochondria? And that's key. That tells you what we call the systemic effect. So outside the cell in circulation, what is this oxidized version? Because this is more stable in blood. So the levels go up, okay? Within 5 minutes, 30 minutes it goes up, 45 minutes is the peak, and by about 2 hours, it's back to baseline. So what the steroid is doing is coming in, it initiates the process, and it's gone. Because it does not want to keep making mitochondria all the time. It only makes it to the point that it is needed. It's like you throw a pebble in a lake. The pebble goes away, the rock goes away, but the ripple continues, right? So the rock comes in, the steroid comes in, starts the process, and it's gone. And that process continues. So when you exercise and you go lift weights, you feel the metabolic changes of that exercise for up to 6 to 8 hours. You've stopped exercising, but that effect continues, okay? But what was also fascinating is in the tissue bed. So if you look on the box to the right, so when you look at the control animals, they have levels in brain. It's 0 because it's below picogram per mil. It's pretty much non-existent. In skeletal muscle, it's pretty much non-existent. In the liver, which is a highly metabolic organ, also used for clearance pathways, there's about 477 change picogram per mil. But look what happened post-exercise. So after 45 minutes, the levels in the brain are up to 25 picogram per mil, and in the skeletal muscle, it's 63. And that's telling you that this thing is made in the leg muscle of these rats who are swimming, but the levels in the brain is going up. Your bioenergetic processes, mitochondrial processes in the brain is going up. That's why when you exercise, you feel good, right? So it's not just one part of the body, it's the systemic part of the body that this thing is going on. Okay, so what have I told you? It's the first mitochondrial autocrine hormone. So this is hydroxypregnenolone. That means it has primary function in the cell that it has been made in, okay? It's released by the mitochondria during increased exercise. It induces biogenesis at picomolar concentrations, shifts mitochondrial fuel preference to lipids or fat. I've shown you all of that data. And it induces expressions of regenerative proteins in muscles and neuronal tissue. I've not shown you the data, but I will allude to that when we speak at the clinical studies. But what happens is the hydroxyprogesterone exists as the oxidized metabolite, and it has the paracrine effect. It has effect on cells other than where the original steroid was made. And does it exist? Yes. This was the first time that anybody has ever reported the existence of the steroid in humans. Okay, so we have now touched the whole story. It's made by exercise, we've seen its pharmacology, and we have now confirmed that it exists in humans. The story won't end there because there's a lot more work to do, and we're excitedly following through. So let's think about, here's the structure, I'm a chemist, structure basis of what it is. So here is a steroid that does this. Now, flavonols are natural products that exist in nature, and they have the molecular footprint of a steroid. The size of a flavonol is like the size of a steroid. So let's look at that. You can overlay this in two different ways. Okay, so if you look at the black ring A and the red ring A, I can overlay each of those two molecules in two different ways. Okay, where AA overlap and AA sit distal to each other. Fair? Okay. Now if I put all the various hydroxys of the steroid in red, okay, this is, and then of the flavonol in black, that's what you get. And let's put what that would imply, okay? It would imply that if this is flavonol, epicatechin has a role, and it can mimic hydroxypregnenolone, there are two ways it could mimic it. It could mimic it as a congener or a structural mimic of 7-alpha hydroxyprogesterone or 11-beta hydroxyprogesterone. Fascinatingly, when we looked at that, the activity was with 11-beta, okay? Hydroxypregnenolone. So that's epicatechin. But what about some related structures, okay? So we looked at epigallocatechin gallate, quercetin, catechin, epicatechin, and a couple more, but let's talk about these 4. And I'll point out that right off the bat, these 3 are not active. The activity only resides with epicatechin, right? And look at the difference, okay? If you look at it, they all have those 5 hydroxy groups of epicatechin. What is the difference? If you look at epigallocatechin gallate, the difference is that it has this thing extra. So instead of the hydroxy, which is green in epicatechin, it has this big bulky group in the red, and then it's an hydroxy group. That's fine. It is structurally reasonably different, so one can say, fine, it's not that active. If you go to quercetin, quercetin has 2 distinct differences from epicatechin. One is the green lobes, and then it has an extra carbonate, okay? So fine, it has differentially there. But look at catechin. The only difference with this thing is how these molecules are oriented. Everything is else the same. So epicatechin's activity, and I'll show you the data on that, is very specific to what it has and how it's oriented in 3-dimensional space. And that tells the chemist that there's a very specific binding that it needs to do. And as I remember, the hydroxypregnenolone I told you about has a very specific binding mode, right? It requires, that's why it's so potent. Epicatechin has similarities to that. But it's not as potent because it doesn't have all the right orientation, but enough to be active, okay? All right, so in the last little bit of time, I think how am I doing on time? I'm all right? Okay, so I'm gonna try and wrap it up in the next few minutes so you guys have time for questions. Let me take you just very quickly to some of the clinical studies we have done. There's many more in vitro and preclinical studies, but I will tell you. So this is a study with Becker's muscular dystrophy patients. They were treated with epicatechin. So I've made a quick switch on you, and so let me remind you what it is. That we were talking about the steroid, and I've told you that epicatechin is a flavonol that mimics the steroid. And now we're studying epicatechin in the clinic, okay? And that is the next set of, part of the talk is touching on epicatechin itself. So they were treated with epicatechin for 8 weeks, and we looked at cardiac function, muscle strength, upper limb function, biomarkers, etc. But the things that I want to really highlight for you is that while we see all the changes on induced biomarkers and etc., the 2 things I want to tell you about is the effect on the muscle and the heart. So these people had cardiac heart failure, and they also had muscle dystrophy. So they couldn't walk well, but they also had a heart function. After 8 weeks, they had physical function, where they were able to bend, pick up sticks, walk up stairs. And we did their muscle biopsy. And this one panel on the left, top and bottom, is before. The top one is higher resolution, the lower one is less resolution. All thing I want to point out to you is if you look at the bottom left, what these striations are called sarcomeres. This is how the muscle transfers force as you work on it. And you can see that those sarcomeres are a little bit thin. On the right, you can see they're becoming thicker. But if you'll notice that I have highlighted in yellow circles, between the left and the right, the number of mitochondria around those have also gone up, okay? So not only is in 8 weeks, the muscle has reorganized to make thicker sarcomeres, but it has more mitochondria. But if you look at the top one, that next part, remember I mentioned to you about the cristae. On the left, I've highlighted those cristae, right? There are fewer cristae. On the right, more cristae. So not only do you have more mitochondria, they're functionally better mitochondria. And this is straight biopsy of human muscle post-treatment of epicatechin for 8 weeks. But the real kicker is what sits in the graph, and that's their cardiac function. If you look at the very bottom, so the solid line, so what we did is we made them exercise. It's called graded exercise. So as they're exercising, we increase the strength of the exercise demand. And from the left to the right, if you see, it's increasing exercise demand. And the panels on the top left is looking at lactic acid. The one on the bottom is looking at blood oxygenation, which tells you how well is the heart functioning. And on the right, top right, is the heart rate. Okay? So, the solid line is before they were on treatment. So if you look at it, the lactic acid, as the exercise goes up, continues to rise. Okay? And if you focus on the top right, the heart rate goes up too. Okay? So that means these guys' heart has to work harder to keep up with the exercise demand, but they're making more lactic acid, so their mitochondrial function is not keeping up with that demand. Remember I told you about the pyruvate and the lactic acid? It's the same story. 8 weeks later, look at that, that has plummeted down. And the key part is the top right. Their heart rate is not going up. That means functionally their heart is much better. It is pumping the blood much better per stroke. Okay? This does not require MRI or anything. It's fundamental equation. Okay? Cardiac output, which is measured by pulse ox, which is, if you look at the bottom left now, okay, they're really maintaining their pulse oxygen levels. The whole part is that when the heart pumps every time, it sends oxygenated blood to the system. It oxygenates it, so the blood is well oxygenated, but the heart doesn't have to beat much. So that's called cardiac output. Okay? If you look at the top left, their lactic acid levels have gone down, so that means their mitochondrial functions have gone up. Okay? And the most important part is that the cardiac output is the multiplier of stroke volume and heart rate. Okay? So if your heart rate is going down, then your stroke volume has to go up to maintain the cardiac output. And the stroke volume can only go up if the heart is better. So what we're seeing is this very, very fascinating data. This is published data, so you can look at it. Then we looked at patients with diabetic cardiomyopathy, and I'll go through this thing. Here we did an extensive study on mitochondrial proteins. This is a 3-month study, 100 milligrams per day, and B is before, A is after, only 5 patients. And this is all this is showing change in mitochondrial electron transport chain proteins compared to baseline. Yeah? And I'm going to go fast. So if you look at it, this is mitochondrial proteins in all 5 patients have gone up. All electron transport chain levels have gone up. Mitochondrial biogenesis has gone up. Modulators of muscle differentiation have gone up. From myostatin is going down, myofollistatin is going up. Dystroglycans levels have gone up, which is very unusual. And here's one I will point out. If you look at this thing about dystrophin. So if you look at 5A, patient 5, before he hardly had any dystrophin, and later he has dystrophin. Why is that important? These patients, after 3 months, who could only go from bed to chair, were walking a mile. But remember, I showed you the Becker muscular dystrophy data. That is dystrophinopathy. That means those patients have a genetic disease that kills their dystrophin. But on the other side, with diabetics, it's their disease that's killing their dystrophin. But the body is bringing back these sarcodystrophin complexes so the body's muscles can function better. Diabetic cardiomyopathy, again, so this is not cardiomyopathy, sorry. This is looking at endothelial function, looking at expressions of nNOS, pNOS, nitric oxide, which has direct association with endothelial function of the vasculature in these patients has all improved statistically. And we see a very significant reduction on B-type natriuretic peptide, which is statistically significant. And BNP is a marker, blood marker for heart failure. And what we're seeing is a very clear reduction in heart failure in these patients. And the most amazing thing was a reduction in what we call beta-galactosidase. Beta-galactosidase is a hallmark for cellular senescence. Okay? What we're seeing is a reduction in markers of cellular senescence. That means these patients are seeing a reversal of somatic systems. Yeah. And they also about up to 20 pounds reduction in weight. And what happens to their sarcomeres? The same. And I don't have to explain to you. You don't need to be a muscle physiologist to understand. Left is before, is after. And you can see the difference. Left is just hamburger mush. The right one is beautifully aligned sarcoglycans. Okay? This is triglycerides, and I know I'm running out of time, so I'm going to go very fast. These are reduction in triglyceride. I give you the statistics. Decreased triglycerides statistically in 30 days. Fasting glucose was down. Fructosamine levels were down. HOMA-IR responses were improved. And these are diabetics. And this is interesting because I told you, remember, that we induce lipid burning, right? Triglycerides are. This is a clinical correlation to the preclinical data we had that you induce lipid burning and triglyceride levels go down. Okay? This is check mark of all the various studies we have done in rodents and humans and the effects we have seen. And I'll wrap it up with the idea that 11-hydroxypregnenolone and 11-hydroxyprogesterone are the autocrine, paracrine, exercise hormones that your body makes when you exercise. But if you cannot exercise, Epicatechin is a mimic that can provide the support you need. This is not for the lazy in us. This is for those who need. So our idea is to promote this in patients and other people who can this thing. And they really share a pleiotropic pharmacological property based on structural relationship. Based on structural relationship that tells you that biochemical processes in your body are incredible, they're powerful, and we should pay good attention to that. And I'll leave you with the idea of the mechanism of Epicatechin, which is unique. And right now we are doing multiple studies already. We've got 6-8 studies going on. So with that, thank you for your attention. Appreciate if you have any questions.
Thanks, Sundeep, for taking us through the complex and yet beautiful mechanism around mitochondria. I've heard this a few times before. A lot of these things I've seen before, but every time I look at the data, the beauty of biological systems just fascinate us more, right? As scientists, as individuals, it's just amazing what happens inside of us on a daily basis. So thank you, Sundeep, for taking us through that. This is 15 plus years of research that you condensed in an hour. So thank you for doing that.
Before you go, I do want to thank a lot of my collaborators, both here at Sphera and also in the US. There have been many, so it's hard to put everybody's credit on that slide. But I have to tell everyone this is not the work I did. It's a work that has been done by many who have collaborated with me. And so they should be gratefully acknowledged. I just have the pleasure and luxury of presenting a lot of their data. So it should be very clear that I'm part of a bigger team that has really worked hard to put all of this information out.
Very rightly said. And I've also had the privilege of interacting with some of those scientists and extremely brilliant set of people across the globe.
Correct. Yeah. And that's why I don't name them, because if I miss a few, then I would not be fair. But I always make sure that they're acknowledged.
No, grateful, Sundeep. Thanks for that. Sundeep, as the theme for today's session is more on how mitochondrial health is implicated in various, disease pathologies or manifestations. And you alluded to this a bit, right from aging to different kinds. So if you could, for the larger audience, articulate the various aspects where mitochondrial health is implicated and can be addressed by exercise, like you rightly said. So everybody gets the message that, guys, please do exercise and eat well. But apart from that, you also said that some people who are unable to exercise, for them also Epicatechin could be a tool. So if your thoughts on that, please.
So that's a wide-ranging question, and we are continuously gathering evidence. I still believe in getting evidence. And so what we are doing is multiple clinical studies in various aspects that leads from primary mitochondrial myopathies to muscle steatosis. I showed you data on muscular dystrophy and triglyceridemia and diabetics, but right now we have studies going on a variety of the other areas. The presumption is that no matter what, either primarily or secondary mitochondria will be affected. Correct? So that now mitochondria regeneration and protection is also fascinating because there's a concept called cellulogenesis. Okay? Cellulogenesis, Professor Navio at UC San Diego promoted that whole idea, which simply means that your body has a capacity to heal itself. It just loses that capacity when the insult becomes chronic, right? So for example, if you break a bone, what happens? You just put a plaster, but the body puts the bone together. You cut yourself, you put a Band-Aid, but the body heals the wound. But now if you have 100 cuts a day, the body is going to start to lose the capacity. Or if you break a bone every hour, your body is not going to heal itself. And that capacity loss is hypothesized, but evidence is accumulating, is related to cellular bioenergetics. That the mitochondria are not able to initiate what we call the cell danger response. Okay? But we're also learning now that the mitochondria communicate from each other. So a cell is under danger, and the cell next to it will sense it, and the mitochondria will go from here to there to what are known as nanotubes to support that cell. So we're still learning a lot, but the whole idea here is the response part to that insult requires transcriptional translation processes to happen that are all bioenergetic and mitochondria dependent. So it's not a big leap of faith to say that if life is not possible without mitochondria, correct, then to sustain health and life, you need mitochondria.
Thank you. I think that's very well articulated. There is also a growing interest in aging and looking aging as a disease. So you did briefly allude to the role of mitochondria in senility or at the cellular level. Can you elaborate a little bit more on that?
Yeah. So as I said, after age 21, you're losing your mitochondria. But the mitochondria is not only being lost because you're not growing, but your environment. The food you eat, the environment you live in, the lifestyle you live in, all has effect on the mitochondria, okay? Now, how it manifests itself is dependent on your genotype, okay? So we are now starting to think about neurodegenerative diseases as metabolic diseases, right?
Yes.
We're thinking about cancer as a metabolic disease. Remember, but if you think about these as metabolic diseases, and the central system that does your metabolism is the mitochondria, then the association is clear. Perhaps the same insult in 3 different people can result in 3 different pathologies. Fair?
Sure.
Because pathology is the expression of the phenotype sitting on your genotype. But the insult is bioenergetics, correct? So if I give, say, lots of sugar to 3 different people, one may develop Alzheimer's, one may develop cancer, and one may develop diabetes. Well, they are 3 distinct diseases, but the underlying insult is the same.
Fascinating.
Yeah.
Yeah.
So that correlation is, I think, pretty well, it's not a big leap to get to that correlation.
I'm constantly being pinged for questions, so I'm going to take some questions from the audience. If there are any questions that you want, Ashish, go for it.
As a child is growing up, till the age of 21, the number of mitochondria is growing in the body, right? But what do we know about the life cycle of mitochondria when it comes to, say, pediatric oncology or any child suffering from muscle atrophy? What's the lifestyle of the mitochondria itself? Because does that growth is uncontrolled? Does that pose any problem?
Yeah, no, of course. When you have insults, and I'll go even to the point of nutrition. I think in our current society, we eat so much processed food and sugar. You don't have to have a disease. You're inducing mitochondrial insult. That's why you see obesity rates are going up. So the idea here is that, yes, so think about it. Your body is growing. It needs mitochondria to grow. The body is providing that. Now, if you put an insult and that mitochondrial level or energetics is required to deal with this insult, it's not doing what it needs to do, which is body growth, right? So there's, because see, also cells have a thing called hormesis, hormetic control, right? So the cells control the mitochondrial level very actively for a few reasons. Number one, that if the cells don't want to make more mitochondria and spend the energy to make the mitochondria if they don't need it, right? Cells are very efficient with their process. They don't waste things. So if you have a child that needs the bioenergetics to grow, and suddenly either through nutrition, lack of exercise, or whatever it is, or genetic things that's going down, then the growth system goes down. Think about a child born with muscular dystrophy, okay? If it's not embryonically lethal, right, the child is born. But the first 6 months, 9 months, they're fine. They're walking around, they're doing things. 9 months, they start to trip. They start to fall. They lose their balance. But if you think about it logically, what happened? Why didn't it happen at the time they were born? And one of the hypotheses is as they're going, what is happening is this dystrophic insult is sapping the mitochondrial energy, and the phenotype, the clinical phenotype, eventually shows up when the internal systems don't have the energy to deal with it, right? And then the body makes choices. It says, I have so much, I'm going to protect the critical functions. I'm going to protect the organs. I'm going to protect the heart. I'm going to protect the brain. And what am I going to let go of? The muscle. But then these kids, as they go in their 14s and 15s, develop cardiomyopathy, because eventually the insult continues and the body loses its capacity to even protect the critical organs.
So it's a related question to that, and I'll take that up. Is children with DMD lose protein every day? Is there a way mitochondria can play a role in it, and can it be balanced? So I know you addressed some of it, but specifically from this perspective.
Yeah, so the rebalancing part is managed by the cell. So what is interesting about this is that if you give the body the resource it needs, it knows what to fix. I'm a scientist, and what I've learned, and this is like the conundrum, right? I finally learned that you don't want to be reductionist, correct? You have to look at the body and the disease in a system.
Systems approach.
But then here comes the mitochondria, because if you were to take a reductionist approach, you can say all these pathologies reduce down to the function of the mitochondria. So coming to your question indirectly, would I expect that rebalancing? Yeah. Whether it has happened in specific cases, it's going on. So what we are doing now is really focused on real-world data and real-world evidence, because the heterogeneity in the population is so huge. So what we're trying to understand is that if we give the system this, are these individuals really balancing themselves to their insult? And what we're seeing right now is yes. So deficits are expressed due to your genotype. Bioenergetics coming can rebalance that phenotype.
So this is a question coming in from our online audience. They're want you to elaborate a little bit on the role of mitochondria and epicatechin in rare diseases in general as a category. So I think that's a bit broader base.
It's a broader base, and I think that's where its key role is. I think, for example, if you look at Leigh syndrome, what do we know? Close to 110 mutations cause Leigh syndrome. Now, if you have to find a gene-based therapy, that's how many you have to create. And that's just Leigh syndrome.
Yeah.
So how do you take monogenic diseases that form these disease bases? But in all of them, the mitochondria play a role. So it's not going to be curative.
Yeah.
Okay? But if you supplement the mitochondria, you will see an effect. And what we have seen is inhibition of progression. Okay? Now, I don't want to claim things that I have no data on, but the data I presented to you today, especially on patients with cardiomyopathy and diabetics, where we actually saw their skeletal muscle get better. These are men in their 60s and 70s, and the muscle I showed you would be in men who would be in their 30s and 40s. Am I seeing regeneration? Perhaps. I'm not going to go ahead and claim that. But the idea here is that can you arrest progression? I think you should be able to.
And you mentioned this point earlier that diseases that are embryonically not lethal, so nature has perhaps decided a way around that deficiency for that embryo to become a viable embryo. Now, coming back to your point about being able to manage the disease rather than going in with a specific intent to curate it from the very basis, can you share any insights from your wide other network of clinicians also as how mitochondrial-based therapies or approaches can help largely manage that?
Yeah, so that's what we said, that it's not curative. I can't fix the gene defect. I can't, if you have diabetes, it may not fix insulin resistance, or maybe, I don't know. But will it help manage the disease? For sure. Okay.
Yeah.
Now, the interesting part about this whole paradigm also is that safety. Okay. So let's touch on that. That when you're going into these patient populations, you have to make sure that what you're giving them is safe.
Yes.
And that is one thing. That's why we know so well about Epicatechin. It's been in human diet for thousands of years. It is safe as can be. Okay. So this is what I have heard. The youngest patient that has taken our supplement is 9 months old, and our oldest is 92. So safety has never been an issue. So that gives you the opportunity to get real-world data and real-world evidence in specific situations and say, hey, what can I know? Because that's how clinicians treat patients.
And that's good, because that leads me to the next question from the audience, from our online audience. And they're asking are there any known side effects of Epicatechin, and how is the dose and duration decided in terms of them starting to see some effect?
Yeah, very good question, because so far we have not seen any adverse events or significant adverse events. What we have seen is that, and this is preclinical, that's why a clinical dose is very clear, it stops working. Okay? If you take too much of it. The other part of our what we have launched is related to the molecule itself, because it's not very stable. It can polymerize easily, it can oxidize easily, it has to be formulated for best bioavailability. Also, remember I showed you some of the other compounds don't have activity. But it's not that they don't have activity. If you have them, they actually inhibit the activity of Epicatechin. So not having them is important, okay? Now, the way we have selected the dose has been through initial preclinical trial and error, modeling into the clinic.
Yeah.
We've done phase 1 clinical studies. It's published in humans. It was done at UC San Francisco, where we did a single ascending and multiple ascending dose. We looked at the exposures and the biomarkers. So remember that green zone I showed you about the ROS.
Yeah.
So think about this, that right area where that ROS exists would be the right amount of steroid it would produce.
Okay.
Correct? And what we're saying is that Epicatechin we're giving is the right amount that sits into that framework. Given that Epicatechin is about 1,000 times less potent than the steroid.
Fair enough.
But still in the druggable potency.
Are there any questions from our audience here? Samantha, go for it.
Going back to patient which saw that there is a significant effect of epicatechin on Becker muscular dystrophy, especially keeping in mind cardiac function and muscle function. Though in case of Becker, we know cognitive, there's not much effect on brain function, like cognitive functions and all. But like in case of Duchenne, we know there is effect on brain also. So what kind of effect have on brain function?
Yeah, we did preclinical studies in aged mice. Unfortunately, they're not good long-term models of dystrophy in animals.
Sure. So Sundeep, just to re-articulate Samantha's question, and I'll try and get it right, Samantha, is that you're asking about the cognitive effect of epi in Duchenne's patients.
So let's look at cognitive function in general, okay? Because while the insult may be different, the consequences are similar, right? The effect on the outcome is similar. Just like in cardiac, I showed you whether the heart failure is from Becker dystrophy, which is primary, secondary on the muscular dystrophy side, or diabetes. Heart failure is heart failure. Now, the brain is a high energy consuming organ, right? It's 2% of your body weight, consumes over 20% of its energy, okay? And it does this all the time. And by the way, a simple fact that it consumes more energy when you're sleeping than you're awake. Okay, just so that you know that when you're sleeping doesn't mean your brain is resting. Actually, it's working even harder. Now, so the energy is the prime force within the brain, correct? Now, what we have seen in our preclinical studies is a very clear effect in cognition in senile animals. So we studied 26-month-old rats, and we see very clear effect on their cognition and the biomarkers associated to neuronal function. We see a great effect on neuronal inflammation, okay? So then we have now limited information from our observational studies in the clinic right now on patients about effects on anxiety, okay, in patients. We are just about to start a study on frontal lobe dementia, okay, which affects 80% of it, which is nothing like simple forgetfulness. I just got up and I can't remember what I was doing 2 hours ago, yeah? And my thought is that the brain should respond quite well and faster. Because if you think about this, in the Becker muscular dystrophy, the muscle goes first, the brain goes next, the heart goes next, but it's the heart that responded first. So yeah, I would think that mental function or effects on the brain I'm very optimistic about that. Yeah.
Another question from our online audience is, can you talk a little bit more about mitochondria and its implication in autoimmune diseases? And is there a correlation?
Is there a correlation? For sure. Okay, because mitochondria control inflammation and immune functions, maturation of immune cells, etc. They all are energy dependent, right? So things just don't happen if they don't have the bioenergetics. Now, we have not directly looked at autoimmune functions, but we do have anecdotal data on patients who have been on this related to induction of autoimmune diseases. Okay. Yeah, I think the data I've seen is that yes, it helps. But again, we have limited data right now, and I think we're continuing to grow on autoimmune disease. We have not specifically studied rheumatoid arthritis, for example, but there are other autoimmune disease functions that we have to look at.
And I know we've hit the time, so I'll take one last question from the online audience, and this seems to be more a Delhi question, that can epi be used for kids, and does it have any implication in respiratory diseases as well?
Yes, it has been used in kids. As I said, Epicatechin we've given to the youngest patient is 9 months old. And there are many other clinicians. Our purpose always is to really work through the clinicians to make sure that they understand what it is it that they should be looking for, or what they can look for. And yes, have we seen effects on respiratory distress? Yes.
Okay, interesting. Very interesting. Sundeep, the questions keep coming through, but I think we have to take a hard stop now. I think this has been a wonderful session. I hope one of many. A
thank
first
you.
of many.
Yeah,
Thank you for taking the time out. It's a pleasure that you're here and that you addressed all of our questions in person.